friends, welcome to Sam Biology class. Friends, today we shall discuss about different types of helical structures that are found in proteins. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Proteins contain three types of helical structures such as alpha helix, 3 by 10 helix and pi helix. First one is alpha helix. Alpha helix is a regular secondary structure. It was discovered by Linus Pauling and Robert Corey in 1951 through model building. In an alpha helix, the peptide backbone is tightly coiled around an imaginary axis. Alpha helices are stabilized by intrachain hydrogen bonds. In particular, the carbonyl group of each amino acid forms a hydrogen bond with the amide group of the amino acid, which is situated 4 residues ahead in the sequence. Thus, for the exception of the amino acids located near the ends of an alpha helix, all main chain CO and NH groups are hydrogen bonded. This figure shows an alpha helix and the blue lines are the hydrogen bonds. These hydrogen bonds are strong, having an optimum distance of 2.8 angstrom. All alpha helices that are found in proteins are essentially right-handed. Alpha helix has 3.6 residues per turn and the pitch of the helix is 5.4 angstrom. What is pitch? Pitch is the distance that the helix rises along the axis per turn. Alpha helices in proteins have an average length of 12 amino acid residues, which corresponds to more than 3 helical turns and a distance of 18 angstrom. This is all about alpha helix. Second one is 3 by 10 helix. A 3 by 10 helix is a type of secondary structure found in proteins and polypeptides. It is the fourth most commonly found protein secondary structure following alpha helices, beta pleated sheet and reverse turns. The 3 by 10 helix constitute nearly 10 to 15% of all helices that are found in protein secondary structures. In such type of helix, each amino acid corresponds to a 120-degree turn in the helix. One complete turn of 3 by 10 helix contains 3 amino acids. So there are 3 amino acid residues per one complete turn. And the pitch of the helix is 6 angstrom. The pitch of a helix is the height of one complete turn measured parallel to the helix axis. So the rise per residue is about 1.3 to 2 angstrom. Like alpha helices and beta pleated sheet, 3 by 10 helix is also stabilized by hydrogen bonding. Most importantly, the amide hydrogen of one amino acid is hydrogen bonded to the carbonyl group of another amino acid 3 residues earlier in the polypeptide chain. This repeated I plus 3 to I hydrogen bonding pattern defines a 3 by 10 helix. Third one is pi helix. It is a right-handed helical structure discovered by Barbara Lowe in 1952. Majority of pi helices are only 7 residues in length. In a pi helix, each amino acid corresponds to a 87-degree turn in the helix. The helix contains 4.4 amino acid residues per turn, and the pitch of the helix is 5.2 angstrom. Like other secondary structures, pi helix is also stabilized by hydrogen bonds. Most importantly, the amide group of an amino acid forms a hydrogen bond with the carbonyl group of the amino acid 5 residues earlier in the polypeptide chain. This repeated hydrogen bonding pattern between amino acid residues I and I plus 5 defines the pi helix. Like alpha helix and beta sheets, pi helix also adapt to regularly repeating dihedral angles phi and psi. Evidences indicate that pi helix can be created by the insertion of a single amino acid into an already existing alpha helix. Thus, alpha helix and pi helix are interconverted by insertion and deletion of an amino acid. So friends, this is about different types of helical structures found in proteins. For more updates, please subscribe my YouTube channel, Sam Biology Class.